This is the revision video for Unit B1, Variation in Inheritance. You will need a pen and some paper for some parts of this video. When directed, pause the video and attempt the question before continuing. We are going to start by thinking about variation. Variation is the differences between organisms. If you look around at all your friends, there will be lots of differences between them, such as different hair colours, different eye colours and different heights. Some of these characteristics are determined by our genes. Some are determined by the environment. Characteristics such as eye colour, nose shape and whether you have dimples and freckles are characteristics that are controlled by your genes. This means these characteristics are inherited. They are passed on in your genes from your parents. Some characteristics are determined by your environment, such as scars, the language you speak and your accent. You cannot speak when you are born. You have to learn by listening to the people around you and copying them, so you learn to speak like them. It is not always as straightforward as this though. A lot of characteristics are determined by a combination of genes and the environment. Things such as mass and height depend on both your genes and the quality of nutrition that you receive. Many scientists have debated for a long time whether genes or the environment are more important in determining certain characteristics, such as intelligence. Most scientists now think that both are equally important and work together. This is known as the nature-nurture debate. All the research gathered in genetic research has shown that genes do play an important role in many aspects of our behaviour and even the way we think, which could explain why it is difficult to change people's attitudes towards things. So why do offspring resemble their parents? We look like our parents because genetic information is passed on to us in the sex cells we developed from. Each human cell, except red blood cells, sperm cells and egg cells, has 46 chromosomes in 23 pairs. 23 that came from the mother and 23 that came from the father. Red blood cells do not have any chromosomes as they do not have a nucleus, and sperm cells and egg cells only have 23 chromosomes rather than 23 pairs, so that when they come together at fertilization, the fertilized egg then has a complete set of 46 chromosomes. Chromosomes are found in the nucleus of cells and are made of a chemical called DNA. Every chromosome carries thousands of genes which determine your characteristics, almost like a map of how your body will be made. So in the nucleus of our cells we have 23 pairs of chromosomes, which are the same in all our cells. These pairs of chromosomes carry information about the same characteristics, and this information is carried on chromosomes in the form of genes. So because we have pairs of chromosomes, we therefore have pairs of genes. This means that there are two genes that control the same characteristic, and the genes that control the same characteristic will be in the same location on each of the two chromosomes. In fact, they are in the same location of the same chromosomes in everybody. For example, the genes that control our eye colour will always be in the same location of the same chromosomes, regardless of the person. But just because we have two copies of every gene does not mean that the two copies are the same. There can be different versions of the same characteristic. For example, one person may have dimples, another may not, but these traits are controlled by genes that occupy the same location on a particular chromosome. These different forms of the same gene are called alleles. Some alleles are dominant, and some are recessive. Dominant alleles will control a characteristic even if they are only present on one of the chromosomes. Recessive alleles will only control a characteristic if they are present on both chromosomes. We can use letters to represent the different alleles. For example, a capital D for dimples, which is the dominant allele, and a lowercase d for no dimples, which is the recessive allele. We always use a capital letter for the dominant allele and a lowercase letter for the recessive allele. Somebody with two dominant alleles would be known as homozygous dominant and would have dimples. Somebody with one dominant and one recessive would be known as heterozygous and would also have dimples. Somebody with two recessive alleles would be known as homozygous recessive and they would not have dimples. The letters that we use to show which genes a person has is known as their genotype. The characteristic that is present as a result of these genes is known as the phenotype. If we look at the 23 pairs of chromosomes that humans have, we actually have 22 pairs of chromosomes, with the final pair known as the sex chromosomes. The sex chromosomes are what determine the gender of an individual. In mammals, females are XX and males are XY. We can use a Punnett square diagram to show why there is a 50-50 chance of a child being male or female. Firstly, we need to identify the mother and father's genotype. As we know that females are all XX, then we know that the mother's genotype must then be XX. And we know that all males are XY, so the father's genotype must be XY. We can then put these into our grid. Now, in the middle of the grid, we combine these genes to see what the possible outcomes are. Now we have our possible outcomes, we can look at the chances of each of these. We can see that 2 out of the 4 are XX, which is female. So there is a 1 in 2 or 50% chance that the child will be female. And that 2 out of the 4 are XY, which is male, meaning that there is a 1 in 2 or 50% chance that the child will be male. This proves that there is in fact a 50-50 chance of a child being a boy or a girl and shows us why. We can also use Punnett square diagrams to show the chances of inheritance for any trait that is controlled by a single gene. But we have to remember that Punnett square diagrams tell us the statistical chance of what will happen when a couple have a baby, but not necessarily what will actually happen. Each time a couple have a baby, there is a 50-50 chance of the child being a boy and an equal chance of it being a girl. But this does not mean that a family will automatically have equal numbers of boys and girls, because ultimately the way that the sperm and eggs combine is entirely down to chance. It is exactly the same when you toss a coin. There is a 50-50 chance of heads or tails each time, but does this mean that there will actually be an equal number of heads and tails in 10 attempts? If you have a coin, pause the video and try it now. Toss a coin 10 times and record what you get each time. Right, let's have a look how we draw a Punnett square for inherited characteristics then. If capital R is for tongue rolling, and lowercase r is for not being able to roll your tongue, draw a Punnett square to show the possible outcomes for a heterozygous mother and a homozygous recessive father. Firstly, we need to identify the genotype of the parents. It tells us in the question that we have a heterozygous mother, so this means that she has one dominant allele and one recessive. It also tells us that the father is homozygous recessive, meaning he has 2 recessive alleles. Now we can put this into the grid and work out the outcomes. This shows us that there is a 50% chance that the child can roll their tongue, and a 50% chance that they can't roll their tongue. When answering a question about inheritance, you should always aim to set out your answer like this. Okay, pause the video and have a go at this one yourself. Right then, we should have identified that both the mother and the father have a dominant and a recessive allele, as they are heterozygous. When we put this into the grid and work out the possibilities, we find that there is a 3 in 4 or 75% chance that their children will have dimples, and a 1 in 4 or 25% chance that they will have a child that doesn't have dimples. The same ideas apply to the way that some diseases are inherited. The ones that you need to know about are red-green colour blindness, sickle cell anemia and cystic fibrosis. Red-green colour blindness is an X-linked disorder. This means that the genes responsible are carried on the X chromosome, and because of this, males are much more likely to be affected. Males only have one copy of the X chromosome, and therefore only need one faulty copy of the gene to suffer with this condition. In the condition sickle cell anemia, the red blood cells produced by the body are the wrong shape and cannot carry enough oxygen around the body. This condition is caused by a faulty recessive allele. See if you can draw a Punnett square diagram to show the chances of two heterozygous parents having a child with sickle cell anemia. Use a capital S for the normal gene and a lowercase s for the faulty recessive gene responsible for sickle cell anemia. Pause the video and have a go. Right then, we should have identified that both the mother and the father have a dominant and a recessive allele as they are heterozygous. When we put this into the grid and work out the possibilities, we find that there is a 3 in 4 or 75% chance that their children will be normal, and a 1 in 4 or 25% chance that they will have a child that has sickle cell anemia. It is actually a little more complicated than that though. People who are heterozygous have something called sickle cell trait. So two parents with sickle cell trait have a 50% chance of having a child with sickle cell trait. The interesting thing is that someone with sickle cell trait has resistance to the malaria parasite. Cystic fibrosis is a condition that affects many organs of the body, including the lungs and digestive system. It causes cell membranes to produce thick secretions that can lead to troublesome coughs, chest infections, and weight loss. Cystic fibrosis is caused by a faulty recessive allele and is inherited from two parents who are heterozygous or carriers. This is because people who are affected with cystic fibrosis cannot have children due to the condition affecting their reproductive organs. Carriers are not affected in any way and probably won't know they are carriers until they have a child with cystic fibrosis. See if you can draw a Punnett square to show how two carriers can have a child with cystic fibrosis. Use a capital C for the normal gene and a lowercase c for the faulty recessive gene that causes cystic fibrosis. Pause the video now and have a go. Right. So heterozygous parents who are carrying the faulty recessive gene have a 1 in 4 or 25% chance of having a normal child, a 2 in 4 or 50% chance of having a child that is a carrier, and a 1 in 4 or 25% chance of having a child with cystic fibrosis.